We're gonna, good evening, everyone. We're gonna wait for 5 minutes so that people can join in. Okay. I guess we can start our session for today. So, what I was planning is, as we are in the fourth week of our particular subject, immunology, I thought our revision will be a better way to see from where we have started, where we are right now, and what else we can expect in the coming days. Okay. And by this, you yourself can, what to say, you can revise your things which you have learned. You can ask doubts if you have any. Okay. So we're gonna start with uh, how we started our particular sessions. So we started basically with the basic introduction to immune system, which included some basic nomenclature, general properties, and components of it. Okay. So if we talk about the nomenclature, so basically the term immunity usually refers to protection against infectious pathogens. Okay. However, reactions to some non-infectious substances, including harmless environmental molecules, tumors, and even one's own molecules, are also considered forms of immunity. It can be, so if it is from environment, which is harmless, but it can create allergy. Tumors, that is the cancerous, our own cell, which are not in our control. And autoimmunity, which means that our own immune system is killing our own cells. Okay. The collection of cells, tissues, and molecules that mediate these reactions is called the immune system. And the coordinated response of these cells and molecules to pathogens and other substances make up an immune response. See, the cells, the tissues, and the small molecules, which all make it happen, which comprises of immunity, they all together are called immune system. And when all of them, the cells, tissues, small molecules, all these come together in a coordinated fashion against particular antigen or pathogen, we call them a immune response. Okay. So till now, what we know is the things which comprises the cells and tissues and the small molecules which act against the antigen in a coordinated manner, it is called an immune response. Okay. So some of the importance of immune system, like why do we need immune system in the first place? Okay, so there are few benefits which is provided by the immune system, and the benefit includes defense against infections. That is, deficient immunity results in increased susceptibility to infections, such as in AIDS, that is acquired immunodeficiency syndrome, right? As the name itself suggests, acquired immunodeficiency syndrome, which means it is a disease or a group of diseases which are a result of deficiency in the immune system. Okay, vaccination boosts immune defense and protects against infections. Defense against tumors, that is, potential for immunotherapy of cancer, control of tissue regeneration and scarring, repair of damaged tissues. So this is a role of immune system. It's not a separate system, right? So if you get a wound, so you want that place to be filled and like filled by the same cells of your body, and it should leave no scar, right? All of us, none of us like basically we like scarring, but we still have scarring. So you can have scars from operations, you can have scars from your basic injury, you can have scars from say your gully cricket, right? So all these things can give you scar, but not all the wounds and the injuries which you have leads to scarring as well, right? Some of them leave scar, but some of them also don't leave any scar. That is also a part of immune system. Okay. Cell injury and pathogenic inflammation. Immune responses are the cause of allergic, autoimmune, and other inflammatory diseases, and for some of the harmful consequences in infections. Recognition of the injury to tissue grafts and newly introduced proteins. That is, immune responses are barrier to transplantation and gene therapy. So, what is a transplantation or a gene therapy? Basically, imagine you are taking something from the outside and your body is saying, no, 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 this is not ours. It's a good thing, right? But it can also be a bad thing. How we have grown as a medical community, as a life science community, as a biotechnology community, now we can challenge some of the diseases and things which we get, right? But one of the challenge still remains that in the transplantation, though we are trying to help the person itself, but the body doesn't know that we are trying to help it, correct? So it will treat, if the matching is not appropriate, it will treat the help as a foreign substance. And the foreign substance will be handled the way our immune response is supposed to be like handling it. Hence, it is like a drawback of our immune system as well as the pro or the con of the pro of the immune system. Okay. So, what is infection and immunity? The most important physiologic function of the immune system is to prevent and eradicate infections. Individuals with defective immune responses are at increased risk for serious, often life threatening infections. Conversely, stimulating immune responses against microbes through vaccination is the most effective method for protecting individuals against infections. This approach has led to the worldwide eradication of smallpox, the only disease that has been eliminated from civilization by human intervention. Okay, so here we can see that the disease and the maximum number of cases in the year recorded in the year, and how it is handled in the, like, this is the most recent data they had of, like, 2019 itself. So diphtheria which had like 2 lakh cases at some point of time, that is 1921, reported only 2 cases in 2019. Same goes with measles, mumps, pertussis, rubella, tetanus, Haemophilus influenza type B, and hepatitis B, right? But look at the polio. So, polio in 1952 reported around 20,000 patients, right? Paralytic patients, which means the patient has impaired movement in the leg or not even properly developed limbs. That case was zero in 2019. And this all happened just because of the vaccination and the uh, spread of vaccination at the proper time to every corner of the world. Okay. Similarly, if we take the most recent pandemic which we faced was COVID-19. So in COVID-19, patients who were not vaccinated, the number of daily deaths, which means if 1 lakh individuals are affected per day, among those 1 lakh individuals who were not vaccinated, around 1.3 of them were dying. Okay. On the other hand, if the person was vaccinated, the average daily death was zero point 0.1. So it's a huge difference, right, in the number itself, correct? So vaccination does help. Vaccination helps us prevent a disease. Vaccination helps our body to adapt to the maybe like uh, in the future, if that particular disease is trying to affect us, it won't be able to affect us that badly, right? So basically, we have trained our own body. Then we talked about stages of host defense, that is, innate and adaptive immunity. So here we started with the definition of immune system. We see what are the roles of immune system. Then we saw what are the benefits of using the immune system information and trying to control diseases. And then now we are entering into immune system. Okay. So stages of host defense, innate and adaptive immunity. Defense against infections is provided by the early reactions of innate immunity and the later, more powerful reactions of adaptive immunity. So, innate immunity, also known as natural or native immunity, present in all healthy individuals at all times. First line of defense, that is, it blocks microbial entry and eliminates invaders rapidly, provides immediate response upon infection, does not require prior exposure to pathogens. Evolutionary older defense mechanism. Now, some pointers about adaptive immunity or specific immunity, or commonly known as acquired immunity. Activated after exposure to microbes, requires proliferation and differentiation of lymphocytes, highly specific to the invading pathogen, involves immunologic memory, that is, stronger response on re exposure, takes a few days to develop, more specialized and potent due to clonal expansion and effector diversity. We'll see this in the coming slides. Okay, so now we see the components of innate and adaptive immunity. First, we saw what is immunity, then we saw what is immune system, then we saw what is an immune response. Now we are seeing what are the components associated with the immunity. So if we talk about the innate immunity part, which is represented here, and the adaptive immunity part, which is represented here. So in innate immunity, we have epithelial barriers. Just below the skin, we have certain cells which are part of innate immunity. Commonly, they are mast cells, phagocytes, which phagocytose the microbe, dendritic cells, which are antigen presenting cells, NK cells and ILCs. And then ILCs basically they are lymphoid cells only. So it can be neutrophil, basophil, all those things. Okay. And then the complement system, which we have not yet discussed. Then, if we talk about adaptive immunity majorly has two components. First, B lymphocyte, and another component is T lymphocyte. So, B lymphocyte gives rise to antibodies, and T lymphocytes give rise to effector T cells. Okay, this is the overview of it. Now we'll see how these cells and components of these immunity or immune system works individually, and then how they work together to come up with the organized immune response. Okay, excuse me. The components of innate immunity, it is the first line of defense. Epithelial barrier, as we have seen in the previous diagram. So epithelial barrier, that is the skin and mucosal tissues, then antimicrobial substances from the epithelial cells. So there are certain, so you know now that we secrete some oils and some other substances on our skin which act as antimicrobial substances. Resident immune cells, that is just under the epithelia, which we just saw, that the mast cells, the lymphocytes, and all those things. Okay. And then what is the function of first line of defense? It basically blocks the entry of the microbe. So the first line is we are not even letting the microbe go inside. We'll see later. But if we completely stop the entry of the microbe itself, which means we don't have to worry much about it because microbe has not yet is in our system, so that our cells should panic and then start the whole immune response. Okay. Then what is the second line of defense? Now imagine, because we are like bombarded with loads of microbes on daily basis, some of them they make their way inside our body, crossing the first line of defense. So then what happens when they enter inside our body? So the first way is phagocytosis or phagocytes. So phagocytes perform phagocytosis. So what are phagocytes? They are neutrophils, macrophages, all these things. Plasma proteins, including the complement system, function. They basically destroy microbes in tissue or in circulation. So the whole purpose of having cells just below the first line of defense is, oh, if you can cross the first line, let's see whether you are able to cross the second line. So, all the components of innate immunity, which are involved in phagocytosis, which are involved in certain proteins, which are taking care of it, or which are basically there to kill whatever is not ours, right? That is the second line of defense. Then, few additional roles are it triggers adaptive immune response, right? So, what it means? It means that to have an organized and controlled immune response, the innate immunity should also inform the adaptive immunity. So then it will be the organized. Otherwise, only innate immunity will know that some foreign material has entered or foreign pathogen has entered, right? So the it is a duty of innate immunity's components to let the adaptive immunity or the components of adaptive immunity know that there is some foreign object has entered our body. Okay. It recognizes limited shared microbial structures. These are called pathogen associated molecular patterns. It is important, right? So basically, how do the second line of defense know that this particular thing is foreign? They don't have eyes, they don't have any sensors that, oh, this doesn't feel like us, right? So, what are those things? These are called PAMs or pathogen associated molecular patterns, which are present on the foreign object which is recognized by the immune system, and then they put it under scrutiny that, oh, we have never seen such thing in our body till today, which means it has to be something from the outside. And hence, then the whole cascade of immune response starts. Okay. The adaptive immune response is mediated by lymphocytes with highly diverse and variable receptors for foreign substances and the products of these cells, such as antibodies and other proteins. Key features of adaptive immunity include, but not limited to, essential for defense against pathogens that evade innate immunity, right? So, adaptive immunity, as mentioned multiple times during the lectures, that adaptive immunity cannot identify the sole pathogen or the PAMS, right? It can only see a pathogen when it is being told by the innate immunity. And when innate immunity will tell the adaptive immunity, it will tell that, oh, this has come, it's already here. We are trying to control it, but we don't know much about it. So we need you to have a very specific reaction or very specific response to against response against to this type of evader or this type of antigen. Okay. Lymphocytes express specific receptors to detect microbial and non-microbial molecules. Recognizes a wide range of antigens. Antigen, what is an antigen? So, antigen is any molecule. It can be a protein, it can be a lipid, it can be a carbohydrate, which can specifically recognized or which can be specifically recognized by the lymphocytes or antibodies. Okay. Generally, they are peptides if it is being recognized by the adaptive immunity. Most likely, it will be a peptide only. Okay. Then, what are the types of adaptive immunity? So, what do we mean by types of adaptive immunity? Till here, we saw that there is innate immunity and there is adaptive immunity. Adaptive immunity only comes into play when things have been evaded from the innate immunity. Okay. Now, even the adaptive immunity, as mentioned earlier, that they are very specific. Okay. So, how does this specificity come into play? And why innate immunity is not so specific? Okay. So, we'll try to understand this in, let's say, with a example which is not exactly our own system or our own body, but some day to day things which we encounter. Okay. Imagine your institute is having an admission spree. Okay. So admissions are going on. Before the admission, so multiple people might have applied in your institute, correct? And then these n number of people or students have applied to different departments. Okay. Now, on the day of admission, at the entrance of your university, how they will be segregated or how they will be treated? They'll be treated in a very non-specific way. Okay. And what do I mean by non-specific way? Because they need to enter the university. The university till here is not concerned whether the students want to go to department A or want to go to department B. They are just concerned whether they have the necessary document to enter into the university. Okay. So they'll just check that. If you have that document, they'll let you pass. But if you don't have that document, they won't let you pass, which means you won't be even allowed to enter into the university far-fetched that you will go reach your department. Okay. So this is an example of innate immunity. So innate immunity is not specific. The guards at the main door of the university are not concerned whether you want to go to department A or B. They are just concerned whether you are eligible to enter into the university. So the innate immune response, what they do is they'll keep searching the space. If the space or the cells or the molecules, they have necessary receptors which are recognized as own, they'll let that cell pass and they won't do anything. Okay. But when other pathogens or other molecules come and they don't have the necessary document to enter inside the university, or say they have document of other university, they won't let it pass. Similarly, if a pathogen comes, they don't have the necessary receptors of self, but they have PAMs, the guards at the university will recognize them and they won't allow to enter, right? So here the non-specificity comes into play. The guard at the main door are not at all concerned whether you are from X department, Z department, Y department, no. Okay. Now coming to the adaptive immunity. Imagine the rush was too much, right? Because every single student is coming on that day for admission. So some innate immunity or some guards were not able to check properly whether this particular student has the document of this university. Okay. So what they do is, by mistake, or say just because of the sheer number of students and intake and human efficiency, right? So they let them pass. Now, this student is inside the university, but it doesn't have the necessary documents to enter inside the enter inside the industry, university. Sorry. So they are basically what they will be characterized as pathogens for our immune response or our immune system. Now, they have crossed the first checkpoint. Somehow they have entered. Now imagine, now they want to go into the particular department because they are inside our system now. They can go anywhere. So now the second checkpoint comes near to when you go towards the academic block of your university. When you go towards the academic block, even to enter the academic block, there will be guards. Okay. Now what will happen is, they now, as the number of students will be reduced, because some of them might go to academic block first, or some of them might go to the residential block first. It completely depends. The number will be reduced, right? So when they are in the academic block region, now again they have to show their necessary documents. Here, first time was a mistake, but second time what happens is, this guard recognizes that this particular student doesn't have the necessary document to enter inside the university. What they'll do, they'll either, what they'll say, ki go back, this is not your place. And imagine if the student is stubborn and it is saying no, 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 I still want to enter. What they'll do, they'll use multiple forces. Either they can call more guards and forcefully the student has to go outside the university, or the student will be allowed to sit, but all of its things such as documents or necessary things will be collected so that it cannot harm the university, right? All these things, this is all imaginary example. Okay. All this thing will happen. Now here comes the role of innate and adaptive. Till here, what will happen? These macrophages will keep circulating. So macrophages are the second guard at the academic block. If the student doesn't have the necessary document, the macrophages will basically come and attack this pathogen and will digest it, will remove the threat. Okay. Other thing can also be done. For example, the pathogen is dealt with the macrophages, but now this guard has a responsibility to tell the headquarters of the guard system, where there are more specific things, that, oh, this entity has entered our university till the academic block, and I have taken control of it, but you should know that it was there. Now imagine while this second guard was dealing with one student who doesn't have the necessary document, another student has entered which also doesn't have necessary document. So what will happen is the first student was already taken care of and was told to the adaptive immunity that such cases are coming. Now, once the adaptive immunity knows that such cases are coming, it becomes specific because now it knows what sort of students to expect. Now, the adaptive immune system will come into play. Okay, so this was just a way of seeing things from a macro perspective so that you can get a better understanding of this. Okay, so the adaptive immunity is itself of two types. First, there is a branch of adaptive immunity called humoral immunity, and the other branch is called cell mediated immunity. Okay, so basically, a humoral immunity and a cell mediated immunity are adaptive immunity itself. It is dependent upon by different cells and molecules that provide defense against microbes at different locations. Okay, so if the pathogen has entered inside the blood, and the branch of adaptive immunity which is dealing of the pathogen in the blood is called a humoral immunity. And if it has entered inside the cell, so the locations are different, right? If it has entered inside the cell, and the branch of adaptive immunity which will be dealing with this pathogen is cell mediated immunity. Okay, so extracellular microbes which survive outside host cells and are readily destroyed when they are ingested by phagocytosis are combated by antibodies. Microbes that have evolved to survive inside host cells, either in phagocytic vesicles or in the cytosol, are eradicated by the action of T lymphocytes. So these are the basic components of humoral immunity. So in humoral immunity, the microbe is of extracellular type. The responding lymphocyte is B lymphocyte. The effector mechanism is basically the use of antibodies. And how antibodies are performing the function? They are preventing the infection and they are eliminating the extracellular microbe by different means, neutralizing, opsonization, complement activation, all these things. Okay. Now, when we talk about cell mediated immunity, so cell mediated immunity, the microbe is of intracellular type. Okay, so the phagocytized microbe, it is inside the macrophage. The intracellular microbe, such as viruses, replicates within the infected cell. The responding lymphocyte is helper T cell and cytotoxic T cell. And the mechanism through which helper T cell helps controlling the microbe is basically helping the phagocytized microbe macrophage, which has phagocytized the microbe, by sending some cytokines on that macrophage, activating it, which will lead to the digestion of the microbe. On the other hand, if the microbe is inside a cell, such as virus, the cytotoxic T cell will come and will kill that particular cell and eliminate the infection. Okay, so these are the two major branches or the way through which adaptive immune system based on the location works. Correct? Now, little bit more in depth or just a textual explanation of humoral and cell mediated adaptive immunity is humoral immunity is mediated by proteins called antibodies, which are produced by cells called B lymphocytes. Secreted antibodies enter the circulation, extracellular tissue fluid, and lumens of mucosal organs, such as gastrointestinal and respiratory tracts. Antibodies defend against microbes present in these locations by preventing them from infecting tissues, cells, and by neutralizing toxins made by the microbe. Antibodies also enhance the uptake of extracellular microbe into phagocytes, resulting in the killing of the pathogen. The addition, in addition, antibodies are transported through the placenta into the fetal circulation and protect the fetus and the newborn from the infections. Some things or few basic details of cell mediated immunity, or what is cell mediated immunity is defense against microbes that have entered host cells, mediated by T lymphocytes, critical for intracellular pathogens such as viruses and bacteria which has been phagocytized. Few limitations of antibodies include prevent microbial entry, but ineffective once microbes enter the host cell. Types of T lymphocytes in cell mediated immunity are helper T cell, commonly called as Th cells, or cytotoxic T lymphocytes or CTL cells. So, helper T cells they produce cytokines. Okay, they activate the phagocytes to destroy ingested microbe in intracellular vesicles. Also recruit phagocytes to infection sites to combat extracellular microbes. Cytotoxic T cells or T lymphocytes, on the other hand, they directly kill infected host cells, including non phagocytic cells, especially effective against viruses and cytoplasmic pathogens. So, immunity may be induced in any individual by infection or vaccination. That is active immunity. Or conferred on an individual by transfer of antibodies from an actively immunized individual. That is the passive immunity. So, what is the major difference between active immunity and passive immunity? So, if you got the COVID-19 vaccine, you developed or you activated the adaptive immunity against the COVID-19 virus once you got the vaccine. You were not infected by the virus by default, but you were given the vaccine. This will be called as active immunity. Now, imagine the antibodies which you formed after your vaccination. I take those antibodies from you and give it to someone who has not been vaccinated or doesn't have the antibody of its own. That will be called as passive immunity. Okay. Now, types of adaptive immunity, as I've mentioned about the active and passive. So, active immunity is developed after exposure to a microbe or its antigens. Individual mounts an immune response and eliminates infection, provides long lasting resistance. Exposed individual becomes immune. Unexposed individuals are still considered naive because they don't have the antibody. Passive immunity is basically antibodies are transferred from an immune system or a individual who has been immunized or produced in the labs. So basically, we are giving antibody from outside. Provides immediate but short-term protection. Does not include immunologic memory because the body itself has not produced that. Okay. Physiological examples. Newborns receive maternal antibodies, that is via placenta and via breast milk. So this is passive immunity. Clinical application of this is immunodeficiency disorder treated with pooled antibodies. So if a person doesn't have its own immune system, we can give this individual antibodies from outside so that it can have a good enough immune response against the day-to-day microbes, though its body is not able to do it on its own. Emergency treatment for infections such as SARS-CoV-2 and snake bites. Monoclonal antibodies used in cancer immunotherapy. T cell transfer to not feasible between unrelated individuals due to rejection. So this is the case of that transplantation and all those things, which has some issue in letting other individuals' antibody go into the another person. So you might have seen this in the current week's lecture where idiotypic, alleliotypic, and all those sort of immunoglobulins were discussed. So some of the properties of adaptive immune response includes specificity, diversity, memory, and non-reactivity to self. So what do we mean by specificity? Specificity ensures that immune responses combat the pathogens or tumors that are encountered. Diversity means, or the significance of diversity feature of adaptive immunity is, enables the immune system to respond to a large variety of antigens. Memory leads to enhanced responses to repeated exposures to the same antigens. Non-reactivity to self prevents injury to the host during responses to foreign antigens. Okay. Now we are getting into the components of what to say components of adaptive immune system. Okay. When we are talking about, say, antibody is a part of humoral immunity. How does antibody come into play? Okay, or how they are produced, because they are specific. So from where this specificity is coming from? Okay, so that can be understood when we see how the antibody is being produced. Because at the end of the day, what is an antibody? An antibody is a protein which is secreted by the immune cells against a particular antigen which has just entered. Okay. So for that, where does this specificity, or where does the thing that the antibody knows that it has to attack to this pathogen only when it is free in the blood, because it is part of humoral immunity, so it will act in the blood, right? So it comes with a process which our body does called clonal selection. Okay. So what is clonal selection? Clonal selection is basically, as I have given you earlier, the example of admission in a university. So what we can say is the student which has entered till the academic block, it was of A type. Okay. Now there is another student, because I told you another student will come at that time, right? So another student will come of Y type, say X and Y. We'll take X and Y here. So the student first which entered the academic block was named X, and the second student which arrived at the academic block was named Y. Okay. So the X student which was there, he had some specific documents, right, which needs to be taken care of by one type of security guard. On the other hand, when the Y student comes, then the documents which is possessed by the second student has another type of document, so it needs another type of guard. So for example, this individual needs to go to the headquarters of the security personnel, and this person can be dealt with the guards without even letting the headquarters know. They can be dealt with the with our normal gate guards. Okay, because it doesn't have like rifle or big guns or small guns. Okay, and this person had big guns, so we need to call the headquarters that CCC, this person has entered. So now the person who, or the headquarters security personnel who is equipped in dealing with a student which has gun is different from the security personnel who is equipped to deal with a person who doesn't have the gun. Okay. So both of them are different. This person cannot deal with a person who has gun, and this person cannot deal with a person who doesn't have gun. Okay. So by this, only such personnel will be selected which are equipped to deal with an antigen which has specific type of PAM. Similarly, this cell will be selected to deal with an antigen which has specific type of PAM. This is called clonal selection. And this is how the activity of an antibody is determined. If it can solve this problem by letting the headquarters know, then it is of anti-X antibody. If the personnel is dealt with the security personnel at the gate only, then this type of antibody is being called as anti-Y type of antibody. Okay, this is how we are achieving the specificity of the adaptive immune system. Okay, some of the other features of adaptive immunity, as we have already gone through this, is just a revision. So they include adaptive immune responses have other characteristics that are important for their functions. Immune responses are specialized, and different responses are designed to defend best against different kinds of microbes and at different sites of infection. So, where were these sites? These sites were blood, that is the humoral adaptive immunity, and the other was cell mediated adaptive immunity when the pathogen is inside the cell. So, for example, there are several classes of secreted antibodies, each of which perform a different set of functions. And there are several subsets of T cells, each of which combat infections in different ways. All immune responses are self limited and decline as the infection is eliminated, allowing the system to return to a resting state, prepared to respond for another infection if it comes. So, what are the cells of adaptive immune system? So, the cells which are producing antibodies, the cells which are termed as T lymphocytes or T helper cells, and the cells which are considered as cytotoxic T cells. So, what are the basic cells which are involved in adaptive immune response? So, first type is called lymphocytes. They are B lymphocytes and T lymphocytes. And then we have myeloid cells. So they are neutrophils, macrophages, dendritic cells, and mast cells. So, what are the functions of lymphocytes? So, they are specific recognition, they are involved in specific recognition of antigens and generation of adaptive immune responses. B cell, basically, they mediate the humoral immunity, and T cells, they basically are involved in the cell mediated adaptive immunity. Talking about myeloid cells, so myeloid cells, such as mentioned here, so they are macrophages mostly. They kill the microbe because remember, adaptive immunity needs the antigen to be processed. The adaptive immunity cannot see the whole microbe, only the innate immunity can see that. Dendritic cells, so they are antigen presenting cells and they initiate the T cell response. And mast cells, they basically secrete inflammatory responses. Okay, excuse me. Now, till here, what we have seen is we have innate immunity, we have adaptive immunity. There are multiple places where adaptive immunity deals with, but both of them are working together. The microbe has just crossed the first line of defense. Okay. Once the microbe has crossed the first line of defense, the macrophages will search for this microbe because they are constantly searching, they're constantly moving here and there. Okay. Then they'll process it. In the same time, they'll also let the adaptive immune system know that this microbe has entered. And then the adaptive immune system will start preparing for this microbe to deal it in a very specific manner. But how does it actually happens on the scale of uh, days of how we perceive things? So imagine if you got cold. Like the season is changing, you might have gotten cold at some point of time. So imagine the person who is sitting beside you was having cold for more than 4 to 5 days, for like basically 3 to 5 days. Okay. And you also got the cold from him because the room was closed and no proper ventilation was there. You were close, you guys were in close proximity. So you also got that infection. Now, what will happen is if you got infection today, that is the 0th day here, what will happen is the B cells which are there, which doesn't know that the infection has entered, will recognize this infection and will suddenly give the antibodies into the blood because this is humoral part of adaptive immunity, remember. Okay. Then it will have an antibody against the infection by second or third day. So you remember when you get cold, first 2 days, like till 2 to 3 day, your symptoms they increase and suddenly then they start to resolve. Okay. So by 0 to 3 days approximately, your primary response is via B cells, which are producing antibodies. So the infection is under control. Okay. Now you felt better after fourth, fifth day. You were okay, ki haan, like your symptoms are reduced. But imagine you again got cold from someone else at this point of time, or some other infection at this point of time. But this infection was also similar to the infection which you just got. So, what will happen at this time, because your body was already exposed to the first infection, it already made memory cells because they have stored that information that this particular type of infection entered, a pathogen entered. Now, once you get this cold or this infection again by say sixth or seventh day, what will happen? The response will be secondary, and now the memory cells will deal. So the time which was taken here to first recognize the microbe, then make antibodies, then secrete antibody and save it into the memory cell, that work has not to be done now. Excuse me. Because the memory cells were already there, the antibodies were already there. Now the pathogen won't be able to survive for more than 1 or 2 days, and you'll get a heightened response because the lag phase is not here anymore. The system already knows this pathogen. It doesn't need any memory of it, and then it will control the infection gradually. Okay, again within 3 to 4, total span will be 3 to 4 days because this is also 3 to 4 days. Okay, this is also around say 7 days primary response. After this, you have the memory cells. Okay, now imagine at the same time when you got the second infection of the same pathogen, because you are roaming here and there, many people are sick, so you can get these infections from anywhere, right? So what will happen? So once your secondary response was occurring for the first infection itself, you receive another infection from the surrounding, but now this is a new infection. So again this new infection will take around 7 days to make memory cells. So for the second infection, this phase will be the primary response. For the second infection which you got again, it will be the secondary response. Okay, this is the basis of how our body deals with the things, and how it is dealing by basically incorporating macrophages, T lymphocytes, B lymphocytes, cytokines, and whatever the components we studied earlier. Okay, then before going to the classes of lymphocytes, as we have already seen, but since the beginning of this class, I am saying that this is a revision so that uh, things can be a little bit clear before moving forward. So we saw this video. We're not gonna see this whole. We're gonna skip some of the parts. But till now, cause things are getting more deeper into the molecular aspect of these, so I just wanted to make sure that you guys are on the same page. Okay. So, so till here, okay, then I'm just checking one thing. So you can obviously see this. Um, I have uploaded it, but the problem which I'm facing right now is that I cannot share the link with you guys, like the document link which you might be getting from the NPTEL itself. So I'll provide you the link, but they are already there on my YouTube channel. Same goes with the slides. Slides are not there. You'll get it later. But this is my YouTube channel. The Himanshi Singh is the name. You can easily search, and you'll get the lectures which I have taken there easily. I'll resolve this problem maybe by the end of this week. So, till here, this video has summarized whatever we have discussed. Okay. So, adaptive immunity, innate immunity, components involved in it, how they are dealing with it, clonal selection, and all those things. Okay. Now we have talked about innate immunity, right? Here we are talking about the cells which are involved in adaptive immunity. Okay. And by adaptive immunity, I mean, what were the components? The components were B lymphocytes and T lymphocytes. In B lymphocytes, we have um, what's B cell receptors. In T lymphocytes, we have T cell receptors. T lymphocytes are of generally two types: T helper cell and cytotoxic T cell. And now you know where they are. Both of them, like T helper and cytotoxic, they are cell mediated adaptive immunity, but they act differently. Okay. So, B cell, whenever it recognizes the antigen, it produces the antibody, which basically either neutralize the microbe, help in phagocytosis, or activate the complement system. Okay. When we are talking about T lymphocytes, specifically the T helper cell, so what they do is they basically help the macrophages which have the bacteria, okay, by encouraging the macrophage to digest that bacteria. How they are doing it? They are doing it by secreting some cytokines. How they are secreting the cytokines? When they see the macrophage which has the microbe inside, they'll recognize it, then they'll secrete some cytokines, and these cytokines, the receptors will be on the macrophages and the associated cells. Okay. So these cytokines will either activate the macrophage to digest the microbe, will start the inflammation response again to kill the microbe, and second, they'll activate the B cells so that now they can produce the antibody and hence help in reducing the infection. Okay. When we talk about cytotoxic T cell or CTL, what it is doing is it is not secreting any cytokine or helping the cells to do the work. It itself is doing the work. So how it is doing that work? It will recognize the cell which has the antigen. and it will secrete some granzymes or porins and all those things, like, which can kill the cell which the cytotoxic T cell just recognized. Okay, and hence it will lead to the killing of the infected cell so that the infection cannot be spread even more. Okay. Then we have regulatory type of T lymphocyte. It basically helps in managing the balance between the activated adaptive immunity and the homeostasis when the microbe was not there. Because you need to control these cells as well, right? So that part is given to regulatory T lymphocytes. Okay. Now, we were talking about the receptors because I just said that TCL is there, T cell receptors, B cells have B cell receptors. So, what are the receptors which are involved in innate immunity, and what are the receptors which are involved in adaptive immunity? So, in innate immunity, you have toll like receptors, TLRs, okay, which recognizes the PAMs, that is pathogen associated microbial. Uh, oh, what was the name? What is the full form of PAMS? I forgot. Pathogen associated microbial patterns. Yeah, microbial patterns. Okay. In adaptive immunity, you have different sort of antibodies. They are also receptors. Okay. When we talk about the type of receptors, as already mentioned, in innate immunity, we have TLRs. There are different types of TLRs. And in adaptive immunity, we have antibodies and we also have T cell receptors because antibodies secreted into the blood and T cell receptors are not secreted, but still they will be called as receptors only. Okay. Then numbers and types of receptors. So about 100 different types of TLRs are there. TLRs means toll-like receptors, and only two type of receptors, that is IG or the immunoglobulin, and the TCR, that is a T cell receptor, is part of the adaptive immunity. Where are they distributed? So basically, they are on the cell. They are membrane bound or inside the cell. Okay. For adaptive immunity, they only express, or the specificity is expressed on the cell membrane. They are not inside the cell. Okay. Then, when we talk about the discrimination of self, non self, both of them, because both of them are part of immunity. So, both of them possess the discrimination ability between the self molecules and the antigen molecules. Then we had a brief discussion about the inflammatory response, how it is helping in controlling the infection. Okay, so we discussed in an inflammatory response that once the infection has entered inside our body, that is the first line of defense has been crossed. The second line of defense will come into play. And the second line of defense was having cells which are roaming here and there, they'll get activated and they'll start recognizing these antigens and then they'll start processing however they were supposed to process. But in the meantime, they'll also inform the other cells or the system, basically our own system, to let them know that something has entered. What this will do is they'll secrete some cytokines or mediators or say small molecules. Okay, this will lead to the increased permeability of blood vessels towards the injured space. That is why it becomes a little bit red, right? Whenever you get some wounds, it becomes red and hot as well. Then once the vessels become dilated and they are more permeable to the cells which are there in the blood. So what will happen when these cytokines are released? The blood vessels will become more permeable, leading to the incoming of particular type of cells, right? And these can be protein molecules as well, such as complement system. It is like basically proteins which are acting against the microbes. There will be cells such as neutrophils, basophils, All these cells will come into the site of wound. At the end, once these cells are there, these cells will do their function. And what will be their function? Because this is still the part of innate immune response. Their function will be to first phagocytize the microbe or the infection and inform the adaptive immune system. Okay, so to reach at this stage, these are the intermediary stages which our system takes. Coming to the, and this again is going a little bit deeper. So we have said here that the cells will come here, it will lead to the digestion of the microbe. So how basically this part is happening, or the sixth seventh part is happening, it is discussed here. So how do the cells know where they have to come, right? Again, as I mentioned, they don't have eyes, they don't know where to go, what to do. They are just completely working on the basis of cytokines and the information which they are receiving from the surrounding. And how they are receiving the information? By cytokines and chemokines, because they all have the receptor for these small molecules, and hence they can see, oh, a type of chemokine is there, then I have to go to this place. Okay, this is how they are basically doing the work. Then we discussed about the innate immune receptors. So in innate immunity, whatever the cells are involved, these cells have receptors. And what are those receptors? Those receptors are called TLRs or PRRs in general. TLR is one of the major class of PRR only, pathogen recognition receptors. Okay. So PRRs are of different types. One major type of PRR, which we have discussed till now or taken its name frequently, is TLR or toll-like receptor. So toll-like receptor is there, C-type lectin receptor is there. In cytosol, these were on membrane. But once we see inside the cells of innate immunity providing cells, we have nod-like receptors, we have rig-like receptors, we have cytosolic DNA sensors or CDS in short, and even inside the organelle, when it is being endocytosed, the TLR will come inside the membrane bound structure, that is the endosome, and there it will also work against the PAMs or recognize the PAMs. Will digest it, present it to the adaptive immunity, that is the T cells, and then the T cell can take any action based on the pathogen. It can either have T helper cell action, or it can have TC or the cytotoxic T cell action. Okay, this is how things are in progress. And again, these are the pathways through which the different PRRs are acting and maintaining the homeostasis of the system, or basically how they are encountering the microbe. Then we little bit started discussing about the recognition of antigen by the adaptive immune system, because microbe is as a whole or via PAM is seen by the uh, innate immunity or innate immune cells. But how adaptive immunity or adaptive immune cells are seeing them? So how it is basically recognizing the antigen, because it cannot see the microbe as whole. So that we discussed little bit. So there what we discussed is B and T lymphocytes express different receptors that recognize antigens, that is membrane bound antibodies on B cells, and T cell receptors on T lymphocytes. Then we started seeing the antigen receptors of the lymphocytes. And here the lymphocyte, we mean majorly the T cell receptors. Okay. So in T cell receptors, we have two types, because the antibody is being secreted again from, like, it's a part of adaptive immunity only, right? And there are receptors which are bound to the cell. And these receptors are called T cell receptors. T cell receptors are not secretory in nature, but B cell receptors are secretory in nature. If you lose the transmembrane domain, they can be secretory. And the secretory B cell receptor is called antibody. This is what we have discussed. So, when we talk about antibody or the B cell receptor, which is secretory in nature, what sort of antigens it is recognizing? It is recognizing macromolecules such as proteins, polysaccharides, lipids, nucleic acids, and some small chemicals. There's conformational and linear epitopes because they are recognizing the antigen. Okay. And then if we talk about the T cell receptors, which is the non-secretory part of T cell, we mainly have peptides displayed via MHC, a major histocompatibility complex. They are again receptors on antigen presenting cells. Okay. And they always have linear epitopes because they are recognizing the processed antigen. They are not recognizing the whole antigen as it is. When we talk about diversity, so in T cell receptor, there is more diversity, while in comparison to antibody, the diversity is still less in comparison to TCRs only. But overall, the diversity for both of them is high. Okay. When we talk about antigen recognition, so in antigen recognition, if you have already gone through the lectures, you know what is a variable region and a heavy chain is there. So you know the antibody, how it will recognize the antigen through which, or how the TCR will basically recognize the antigen. Okay, so this is where we were heading towards and how we concluded our lectures last. So then we saw some of the B cell receptors. What are the properties of these B cell receptors? So they are membrane bound antibodies. They recognize diverse chemical structures such as proteins, lipids, carbohydrate, nucleic acid, and small molecules because they are also antigen presenting cells. So they have to see the molecule as it is. Detect native shapes, conformations of molecules. Activated B cells secrete antibodies with identical specificity. Okay. The functions of B cell receptors include they bind or eliminate microbes and toxins in humoral immunity. T cell receptors, if we go through them, so T cell receptors, they only recognizes peptides which are presented by MHC molecules or MHC proteins or MHC receptors. Okay. Antigens are from intracellular proteins or extracellular proteins processed by APCs or the antigen presenting cells. They do not bind to free or soluble antigens because they cannot see that antigen. Okay. Functions of T cell receptors include they are target oriented and they can kill cells which are presenting the antigen. You know, remember, right? If antibody is there, then also it will lead to the cell death. If TC is activated, then also the cell will be killed. If TH is activated, then also the cell will be killed. Okay. Then we started going into the structure of these antigen receptors or T cell receptors. Okay. So overall, the organizations of regions in an antibody or antigen receptors have two major regions. One is variable region and the other is a constant region. So the variable region, the antigen recognition, they differ between lymphocyte clones. Okay, which means if A type, you remember the example which I showed you earlier in clonal selection, in A type of cell, anti A type of receptor will be there. Excuse me. In B cell, anti B type of receptor will be there. So both the antigens will be recognized by different cells which are having anti-A and anti-B receptors. Okay, and that region will be variable region of the antibody or the receptor. When we talk about constant region, so they are they provide the structural integrity and binding to signaling molecules conserved across clones. What do we mean by this? We mean, okay, let me go back a little. This one. So now if you see here, both of them look quite similar, right? The distinguishment is only in the tip of these. This is curved and this is angular. Okay. So the curved part is the variable part because it is recognizing the curved microbe. The angular part is the variable part which is recognizing the angular antigen. But the tail of them or the branch of them looks quite similar, right? So they are the constant regions. But though they are coming from different cells, right? This is a different antibody. This is a different antibody. The major change lies in the antigen recognition site here, and the constant region is constant because they are immunoglobulins. Both of them are immunoglobulins, right? Ig family. Then, if we talk now from where this variability is coming, because at the end of the day, they all are proteins, right? So, the variability will come from the change in the amino acid sequence. If they're conserved between two antibodies, which means they are close together. If the sequence of the protein or the amino acids is different between two proteins, which means they're coming from different source. Okay. So if we talk about the sequence variation, the variation in the antibody or antigen receptor is present in the variable region. Even in the variable region, the region which is highly sequential varied is called hypervariable regions or complementarity determining regions or CDRs. Okay. CDRs, they are the key specificity, which means cell 1, which was anti-X, the sequence which is changed between anti-X and anti-Y will mostly be from the variable region, and even in the variable region, a place will be highly variable called hypervariable regions or CDRs. Okay. Now, if we talk about the functional design. So variations limited to small regions, they maximize antigen binding diversity while maintaining receptors. So it's pretty basic, right? Then we started going into the signaling of it. So this is where we were supposed to discuss our onward classes. Okay. So till the last slide, this was the overall discussion we had till now with respect to the immunity. What is immunity, what are the components of immunity, how they are dealing with dealing with things, dealing with pathogens at different places at different time, what is the time lag between the primary response and the secondary response, what are the components which are involved in primary response and secondary response. This was an overview of it. Then we started getting into the structure of the components which are doing this. Okay. And few structures which we are discussing is the antigen receptors. Antigen receptors are on B cells and on T cells. Till now, we saw about the antibody, which is the secreted B cell receptor. Okay. Now, if we talk about the signaling, because receptor is there, something is bound, something is identified. Now, what happens in the downstream of it? That we're gonna see. So, the association with invariant proteins. So, antigen receptor polypeptides pair with invariant membrane proteins. They deliver intracellular signals after antigen recognition. Variability in receptors, signal transduction via conserved proteins. What are the complexes involved in this? It is the B cell complex and the T cell complex. Till now, we studied about B cell receptor and T cell receptor. But for the cell to perform its function, or the downstream pathways and the cascade of biochemical reactions which needs to be taken after the cell has recognized something, it doesn't only happen because it has recognized a B cell receptor. The B cell receptor will have other co-receptors along with it, which needs to be taken care of. Then only the cascade of events can take place. Okay. And what is the same goes with the T cell receptor. So the mechanism of signaling which happens is antigen binds extracellular portion of the receptor because the receptors are on the surface. Associated signaling proteins phosphorylated on tyrosine residue by protein tyrosine kinases. This is one way of signaling. Okay. It initiates signaling cascade then leads to the gene transcription, then the protein is produced and the lymphocyte response, or basically the response which the cell has to do, whether it has to secrete cytokines, whether it has to engulf the microbe, whether it has to endosymbiont the microbe. All this will depend upon the cell's internal signals. And these signals initiated because the surface receptor recognized something. Okay, and here something, it can be a pathogen as well as it can be a MHC molecule which has the pathogen. Okay, so before going into the deep details of T cell receptors and B cell receptors, this is a brief overview of these receptors. So let me just play this for you guys. भैया टीवी थोड़ी देर के लिए बंद कर दीजिएगा, लाइट थोड़ा समय हो गया गए हुए. Excuse This was the very basic of what we have discussed till now. From here onwards, we're gonna see the structure of antibodies, how they are different, what are different sorts of antibodies are there, how they are different, and what are the problems associated with different antibodies, same antibodies, all those things in the next class. Okay, and then we'll discuss some questions as well. Today, I'm skipping the questions because very few of you are present here today. So I want you guys to have the exposure to questions. It will help you in your assignments as well as in the exam. So I'm skipping questions today. So hope to see you all the next time, right? So we'll discuss some questions and then we'll cover some new topics as well. So yeah, thank you so much. This is all from my side.